Okay, so just an introduction. Our project was a journal entry to we haven't decided what journal yet, but the project itself is complete, so all that we're working on is just figuring out where to submit it to. Our project is it's a research paper on CRISPR gene germline editing, and it basically goes into the depths, it goes into ethical issues, and basically a general overview of what CRISPR is into one paper. Okay. Shrinika, do you want to start?
Yeah. My name is Shrinika, and I'm going to be talking about CRISPR germline editing. If you don't know what CRISPR is, it stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's a gene editing tool that works kind of like a pair of molecular scissors. Scientists use a guide RNA to bring an enzyme called Cas9 to a specific spot in DNA where it makes a cut. From there, the DNA can be disabled, corrected, or have new genetic material inserted. CRISPR is already being used in medicine to treat things like genetic blindness and certain cancers. It is also being tested in agriculture to make stronger crops and even in conservation biology. For example, scientists have explored editing Asian elephant DNA with woolly mammoth genes to possibly bring that species back. CRISPR was first noticed in 1987 in bacteria when researchers saw unusual repeating DNA patterns. Later, in the 2000s, scientists discovered that CRISPR is part of how bacteria defended themselves against viruses. They save bits of viral DNA so they can recognize and fight the virus if it comes back. The big breakthrough came in 2012 when Jennifer Doudna and Emmanuel Charpentier figured out how to turn CRISPR-Cas9 into a gene editing tool. In 2013, scientists edited human cells with it for the first time, and since then the technology has grown quickly. In fact, in 2020, Doudna and Charpentier won the Nobel Prize in Chemistry for this work. I don't The image on the screen is a timeline of CRISPR. It begins in 1933 with the discovery of CRISPR sequences in archaea and moves through major breakthroughs like the identification of Cas9 in 2005, its adaptation to human cells in 2011, and the first published studies in 2013. The timeline also highlights controversies, like the gene-edited babies in 2018, as well as a recognition with the 2020 Nobel Prize. Most recently, in 2023, CRISPR reached a major milestone with its first successful phase 3 clinical trial, showing its potential in real-world medicine. Now on to germline editing. Germline cells are eggs, sperms, or embryos. One major example is from a biotech company called Colossal Biosciences. They used CRISPR and cloning techniques to insert genes from the extinct dire wolf into modern gray wolves, creating pups with dire wolf traits. They hope to expand this to other extinct animals like the woolly mammoth. While exciting, this shows how powerful germline editing can be. But with that power comes big ethical questions. Germline editing is risky. CRISPR can sometimes cut the wrong spot in DNA, causing mutations that could be harmful later on. There's also the issue of consent, because embryos can't choose to be edited, yet those changes affect them and every generation after them. Beyond safety, there are concerns about fairness. Some worry that gene editing could not be used just to prevent diseases, but to create so-called designer babies with select traits like intelligence or appearance. If only wealthy families can afford these enchantments, it could only make social inequality even worse. Because of these concerns, most scientists and ethicists say germline editing in humans shouldn't be used right now. Instead, they call for more research, strict regulations, and open conversation between scientists, lawmakers, and the public. Some argue it could be justified in rare cases, like preventing a deadly inherited disease where no other treatment works. But even then, it would require global agreement and very strong safeguards. In conclusion, CRISPR is an incredible tool with the potential to change medicine, conservation, and even our understanding of life. But when it comes to germline editing, the risks, ethical challenges, and long-term consequences mean that we have to move forward very carefully. Now, Kevin is going to talk about the limits and risks of CRISPR.
I'm Kevin, and I'll start with the limits and risks of CRISPR. So, our limits and risk paragraph talks about the limits and risks of using CRISPR as a gene editing tool, and it shows that it has a great potential but it comes with important challenges. It explains that beside ethical concerns, off-target effects can happen, where DNA is changed in the wrong place, leading to harmful proteins and immune reactions that may even cause strokes or heart attacks. The paragraph also mentions on-target effects, where edits at the correct spot can still create big deletions, chromosomal problems, or cell death, sometimes resulting in tumors. Finally, it says that while scientists have made CRISPR more accurate, the safety issues described here are still limited because of how widely it can be used. Leading on to the next section, which is policies and regulations, this paragraph talks about how different countries regulate CRISPR and the human genome editing, and it focuses on the US, UK, and China. It explains that the US is a very cautious country restricting embryo editing and reproductive uses, whereas the UK allows research under the Human Fertilization and Embryology Authority as long as the embryos are destroyed within 14 days. This paragraph also mentions China, which tightened its laws after the 2018 CRISPR baby scandal, where a Chinese scientist edited gene created gene-edited babies. They were twins, but that went against China's policies, and it still struggles with enforcement and transparency. Other countries like China, Germany, and Canada keep strict bans, and Japan and South Korea allow limited research only. Overall, this section shows that global views on CRISPR are divided. Next, we have a table, and the table is basically the previous paragraph, but split into more simple terms. It shows comparisons between China, the UK, and the World Health Organization, and it shows what they allow versus what they don't, which limits or which limits the research of certain countries compared to others. Our next section is CRISPR applications, and this paragraph talks about how CRISPR has moved from lab research to real-world clinical therapies, where many trials now focus on serious genetic disease. It explains that as of 2024, CRISPR is being used to treat blood disorders like sickle cell disease and thalassemia, with exa-cell showing lasting benefits in trials. The text also mentions new uses such as cancer immunotherapies, treatments for inherited eye conditions, and research on metabolic, lung, and neurodivergent disorders. Overall, this paragraph shows that CRISPR medical applications are growing quickly, marking a major shift in the treatment of gene diseases. Our last paragraph is the reflection and today's state of germline editing, and this paragraph talks about the remaining questions and concerns surrounding CRISPR, especially about ethics and long-term safety. It raises issues such as whether it's right to use CRISPR on viruses without knowing future effects, if scientists should edit human genes, when challenges might pass to future generations, and how to prevent misuse. The text explains that although germline editing is done only in laboratories, a 10-year global moratorium was proposed in 2025 because of safety risks, off-target mutations, and ethical uncertainty. Overall, this paragraph shows that while CRISPR research is advancing, clinical germline editing is still considered too risky and heavily restricted in many countries. Our resolution is that taken together, the previous sections show that CRISPR offers remarkable clinical potential, yet its limits and risks and uneven global regulations demand caution. Moving forward, careful research, international cooperation, and strong ethical standards will be essential to ensure CRISPR develops safely and responsibly. And these are our sources. Thank you.
Thank you so much. It's wonderful. It looks like your research paper is almost ready to be published, and you guys did very wonderful job. I really love where you have the limitations and what could be like the potential threat for the future, or if we are not very careful with the CRISPR editing. So thanks very much. Thank you very much. And just for the sake of the time, I'm not going to allow any question.